origin of replication, where the replication of the desired DNA fragment starts. And a selectable marker is helps in identifying and eliminating non-transformants and selecting permitting the growth of the transformants they have a recombinant DNA. Okay, now if the recombinant DNA, example this plasmid, if it's in the region of ampicillin resistant genes, if it's in if it inserted, like some genes, desired genes is inserted, then it lose the resistance to ampicillin. That's it. So this is how you identify like recombinant recombinants from non-recombinants. Okay, that's it. And how to identify transformants and non-transformants. If they do not if you place them on like ampicillin and tetracycline, if they show the resistance, then they are transformants, and if they do not, they are non-transformants. So this is it. Now another method is insertional inactivation. So if you insert something in the coding sequence of an enzyme beta galactosidase, so this is insertional inactivation. Now they do not produce color, and the color is blue, right? That's it. You see, presence of insert results into insertional inactivation of the beta galactosidase gene, and the colonies do not produce any color. These are identified as recombinant colonies, right? Now like vectors for cloning genes in plants and animals. So in plants it is Agrobacterium tumefaciens. Okay, in animals it is retrovirus. Now this retrovirus can cause the cancer, so they are disarmed before they are used as vectors. Okay, now in plants, as I told you, that it is Agrobacterium tumefaciens. It causes tumor, so it's DNA, that is tDNA, causing tumor. Now okay, so okay, now the tumor inducing TI plasmid of Agrobacterium has now been modified into a cloning vector, which is no more pathogenic to plants, but is still able to use the mechanism to deliver genes of our interest into a variety of plants. Okay, now that's it. There is like competent host, like how to transfer the recombinant DNA into the cell. Now you see, the simple method is like plant have like cell wall. So now okay, bacteria, how they will take up DNA, like first put it in the divalent cation, then put it on an ice, then heat shock like 42 degrees Celsius, and then again put on ice. So this is how bacteria takes up the recombinant DNA. Now in plants it is high velocity microparticles of gold and tungsten coated with DNA. It is biolistic or gene gun. Okay, now disarm pathogen in case of like animals, and in plants, in animals as well, micro injection. Okay, micro injection. Now that's it. Another like lysozyme, that is bacteria, cellulase, plant cells, chitinase, fungus. So these are like kind of enzymes that cause the degradation. That's it. Now for RNA it is RNase, and for protein it is protease. That's it. Now another main thing, like how to separate the DNA. You put it in the isolated, chilled ethanol, and the process is spooling. That's it. Okay, anode is positive, right? And one more thing, remember that like both the vector and desired DNA is cut by the same restriction enzyme, so they produce, so it is easy for the ligase enzyme to join them. Okay, now eco RI1, its palindromic sequence is GAA-TTC, GAA-TTC, 5' to 3', 5' to 3'. Now that's it. Now you see like uh, nucleases has two kinds like exonucleases and endonucleases. Now exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make cuts at specific position within the DNA. That is why exonuclease and endonuclease. And what about restriction endonuclease? So these are kind of like special. They work by functions by inspecting the length of the DNA sequence. Once it finds a specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbones. Okay. Now each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Okay, that's it. That's it, right? Now uh, it's a like you see. Okay, what about the restriction? Look, first restriction endonuclease enzyme is HIND2. Okay, it is not EcoRY, it is HIND2. Now remember this. Now how? Okay, naming is not that important, but now what about plasmid? It is autonomously replicating circular extra chromosomal DNA, and plasmid is also like kind of double standard, but it is circular, right? It is extra chromosomal DNA. It's like it has ability to divide without like the genomic, with the help of main DNA, right? And how first rDNA is made using the native plasmid of Salmonella typhimurium. It is a bacteria. Now by Stanley Cohen and Herbert Boyer in 1972 year. Okay, now now they put the antibiotic resistant gene into them. That's it. Now this is possible. How recombinant DNA technology is possible is because of molecular scissors, that is restriction enzymes, very enzyme which we study. The special one is restriction endonuclease, right? Now vectors, they transfer the piece of DNA. It's kind of like carrier or kind of vehicle. That's it. Now that's it. Now DNA ligase, they join the DNA fragments. That's it. Now cloning, they make the multiple copies. That's it. And the techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning and gene transfer. So gene cloning, then gene transfer, by the vectors. Okay, when vectors are put it into the host, there is gene cloning and recombinant DNA using the restriction endonuclease. That's it. Origin of replication, where the initiation of replication starts. Okay, now like biotechnology, they contain like genetic engineering, bioprocessing. What is genetic engineering? It is technique to alter the chemistry of genetic material, both DNA and RNA, to introduce this into host organisms and thus change the phenotype of the host organism. Now bioprocess engineering, it is kind of like maintenance of sterile microbial contamination free ambience in chemical engineering process to enable growth of only the desired microbe eukaryotic cell in the large quantities for the manufacture of biotechnology products like antibiotics, vaccines, and enzymes. Also biotechnology is useful in like production of like how many, like in vitro fertilization leading to test tube baby synthesis, gene using it like gene therapy, and also developing your DNA vaccine, correcting defective gene, default gene. All these are like part of the biotechnology. That's it. And that's it. And what about like the definition of biotechnology? It is the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. So this is like biotechnology. And in biotechnology, it's not about like biosynthesis, but is always, but it's also about like the marketing, right? Marketing the product. We study in like downstream processing, right? So this is it. Okay. Okay, what are the sticky ends? Like the sticky ends, you see in the example like eco ri, it cut like between the G and A, the same sides. Okay, both the vector and foreign DNA. Okay, you see it's also written like somewhere else, like that line. Okay, where is it? Like, okay, Mhm. Okay, you may have realized that normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. So, unless one cuts the vector and the source DNA, that is the source DNA, that is, we want the desired DNA from these, and the vector that is required for like the cloning, right? The transfer. So, with the same restriction enzyme, the recombinant vector molecule cannot be created. So, that's it. And yeah, that's it. Now you see, like I told you, like a recombinant DNA molecule is transformed into the E. coli cells. If they are transformed, then they are, then they are like transformers, and if they are not, they are no non-transformers. That's it. And this is it. Okay.